Okay, welcome everybody. Welcome to our webinar, Turning Your Center into a Precision Brain Medicine Institution. My name is Brad Fernald. I'm Vice President of Omniscient Neurotechnology. We're a technology company focused on delivering practical and intuitive software solutions to connect pioneers in neurosurgery, neurology, and neuroscience with subject-specific brain analytics. So our goal is to revolutionize brain care with data, and we're pleased to bring you this webinar. So throughout this webinar, we hope to bring you practical insights, into the role and potential of Connectomics as a validated brain fingerprinting technique. And that's for the use in research today, as well as the future potential clinical use of this technology. Practical information on how to implement data-driven in-hospital neuro studies, as well as real case studies on the use of this approach. And today we're joined by three leading clinical researchers in this space. We have Dr. Michael McDermott, who is a neurosurgeon and chief medical executive of Miami Neuroscience Institute. He's a professor of neurosurgery and translational medicine and chair of the division of neuroscience at the Herbert Wytham College of Medicine at Florida International University. And prior to joining the team in Miami in March of this year, Dr. McDermott served as professor of neurosurgery at UCSF, spanning nearly 30 years. Dr. McDermott is world-renowned in the surgical management of meningiomas, but beyond that, as a premier educator, researcher, and engineer. We have Dr. Michael Rosenbloom, who is a behavioral neurologist based at the Health Partners Center for Memory and Aging in St. Paul, Minnesota, where he serves as the clinical director and chair for the Department of Neurology. He attended medical school at Columbia and received his neurology training at the Harvard program at Mass General and Brigham Women's Hospitals, and completed a behavioral neurology fellowship at UCSF, focusing on Alzheimer's disease, as well as atypical dementias such as frontotemporal and rapidly progressing dementia. Dr. Rosenbloom's research includes imaging and focal presentation of Alzheimer's disease, intranasal therapeutics for treatment of central nervous system disorders, and early detection of Alzheimer's disease, and the application of resting state fMRI to Alzheimer's disease in clinical practice. And lastly, we have Dr. Michael Sughrue, who is a neurosurgeon at Prince of Wales Hospital in Sydney, Australia, and Chief Medical Officer of Omniscient Neurotechnologies. Prior to joining the team in Sydney, Dr. Sughrue built one of the busiest brain tumor programs in the US at the University of Oklahoma, where he dedicated himself to being an accomplished researcher, having published over 200 peer-reviewed scientific publications in medical journals and having made significant contributions to the field of connectomics. He trained in neurosurgery at UCSF and is a believer that despite amazing advances in computer science over the last few decades, doctors still make decisions based on old methods, and that we can only progress by harnessing technology to provide better information to doctors. So before I hand it over to Dr. Sughrue, I'd like to address some housekeeping items. You may submit questions throughout the webinar using the Q&A tab in the Zoom interface. The button is highlighted here. We'll monitor those questions, and there'll be a Q&A session at the end. We'll open up the floor at that point, and you can also submit questions at the end as well. The Q&A session may run over on time, but members of our team will stick around and make sure they answered all the questions. And throughout the webinar, you may be presented with poll questions. We'll go ahead and launch the first one now. And these poll questions help the speakers understand a bit more about the audience. So we'll go ahead and launch that poll question. And just go ahead and choose your answer and submit it, and that's all you need to do. Great, and thank you. And with that, I'd like to hand it over to our first speaker, Dr. Sughrue.
Hi, everyone, and thank you for coming. I'll share my screen. I have to say I'm impressed because I'm fairly certain Brad was reading my biography without actually looking at a piece of paper, which is amazing to me. Today we're going to talk about Infinitome, and I called it the Infinitome concept. One of the things that's interesting about cloud platforms is that it's a constant state of evolution. We're constantly evolving what Infinitome means, what it's built into, and again, it will constantly improve as time goes on, because if nothing else, we're making better machine learning tools based on data. So ultimately, I would hope anyone who's watching a call at this hour about brain connectivity probably is aware that brain disease is an extremely data-intensive problem. We all know that the brain is extremely complex. It has billions of connections in very complex ways that cause very complex signals, characteristics, and communication patterns between areas. And in order for us to understand this, we need to really integrate intelligently multiple forms of information, such as MRI of various kinds, clinical data, neuropsychological data, neurological data, disease state data, etc. And anyone who's really has experience with pretty much any of the diseases knows that there's a diverse collection of symptoms. No patients have the same exact symptom profile. And while we're often recognizing patterns, there's significant differences between patients because if nothing else, this is happening in the body, which is even more complicated because the brain is a subset of the body. And when we try to tackle these problems, which are extremely high dimensional and difficult, we're doing this in a very complex, highly regulated medical and legal environment. And this causes a whole nother set of issues that works against us in many times being able to leverage big data in a way that, say, a bank would. So what we're going to, Infinitome's fundamental core base piece is based on connectomics. And this comes from a term that is a play on the word genomics. So genomics are big data for genes, and connectomics are big data analytic approaches for all of the connections of the brain. And there are multiple types of connectomes you can create. Functional is one type of multiple forms in that. And ultimately, it's almost become a field to itself. And this is noted by the fact that there was a large-scale project in the Brain Initiative that called the Human Connectome Project, and many people have spent a large amount of time working to annotate and work further on a lot of the high-quality data that was produced in that project. And so, again, the field has taken a quantum leap, and one of the biggest drivers is the ability and innovations in large-scale, enterprise-grade computing solutions, most notably things like machine learning at large scale. So what Infinitome is, ultimately, is a practical application linking some of the technology from the computer world and some of the approaches that people do in the industry, for example, at some place like Amazon, Apple, Facebook, etc., to bring large-scale machine learning, but built around the brain, which has unique challenges, most notably that it isn't just a numbers problem, it matters what part of the brain you're talking about, and there's a massive amount of prior knowledge necessary to leverage this. But we utilize techniques such as cloud-based machine learning, federated machine learning, and work to integrate data with the existing IT systems of a hospital. It's able to work with PACS and also electronic medical record systems such as Epic, and to allow people to organize huge data sets and to work quickly to identify patients of interest without having someone do this manually. And obviously there's a cost scaling issue involved in this. It allows us to really look at medical records in a way that we haven't done before. Instead of having individuals go through it, we utilize things like natural language processing. And we link this to automated data collection systems, most notably neuropsychology, which I'll talk about briefly. And eventually with the goal of getting all this data in a form that can be used to mine machine learning data models at scale. Now, at the base of this is the Infinitome viewer. Now, there's a lot of ways you can repurpose this. And we have other products that are in different stages of development that are based around this architecture. But what you can see is this is a 3D visualization device. It works in Google Chrome and leverages things like diffusion tractography, in this case, constrained circle deconvolution, and machine learning to provide insight on resting state fMRI and DTI in a way that ultimately is designed to be clinically actionable. And you can see many of the component parts. But it's important to note that there's other parts of Infinitome that are not directly in the viewer. The viewer is kind of a way to interact with the data in clinically applicable pipelines, and I believe some of the other speakers will talk a bit to this. But there's a lot of other things that are in evolution or behind the scenes that are coming out. And where the concept came from, what is the Infinitome concept? And I actually have to credit my former chairman at the University of Oklahoma, Tim Mapstone, for first pointing this out to me, that we have one of the greatest neuroscience resources in the world, which is we have large collections of neurologically sick people generating enormous amounts of data in our hospitals. But because hospitals weren't designed with research in mind, they're not very friendly and easy places to collect data. And as a result, most of the data just stays inside of an internal loop. And very, very little and very, very few of these patients end up in a study where we can learn about their disease and help other people with it. And what this means is that our studies are inherently underpowered and variable, so to speak. We don't collect all the data possible just because there's a limited amount of resources. You know, it's hard to get $30 million grants that you could do whatever you want. And so part of Infinitome is actually allowing us to really harness what's on the left and make it so that what's on the right and what's on the left are really kind of the same place. So to that end, we focus mainly on individual level data and clinically achievable scans. We're not aimed to become the world's leader on processing 120 directional DTI. We don't think that's ever going to be practical or scalable in clinical practice without a major evolution in MRIs. If that happens, then we're happy to modify our software to address this. But we really made our algorithms robust so that they could collect data in the real world. And what you see is diffusion tractography and resting state fMRI. Not that TASC doesn't have a utility in this, in research, but it's not something we could do in the hospital very easily. And most people who've attempted to get test-based fMRIs know the pain of this. And what we do is we turn really sophisticated algorithms, and we're always working to develop new ones as the literature evolves and our understanding evolves, to really extract large amounts of data for these data sets. One of the DTI makes beautiful pictures in part because it's extremely complex and we're just amazed at our brains. But that's not practical. And what we really need to be able to do is to tackle it at the level of complexity that it actually presents. And this is something like here, which is a very giant matrix. But scans by themselves are only so interesting. And so you need to know who these people are. What are their neurological problems? What are their confounding variables? And we leverage this by going to the medical records with natural language processing and also linking it to neuropsychology in a way that when they all hit the cloud, they're in an organized way that we can utilize workflows. You can see in the upper right, this is something we're rolling out over the next few weeks, to be able to take a huge data set and quickly identify who you want to be studying. Sending this to an enterprise grade machine learning workbench. Again, this is something that will be appearing over the next few months. And eventually eliminating the barriers to working across centers and across nations to do massive scale projects that are really aimed at the level of complexity that we need to be able to address this to make a difference. So none of these are new. Like we didn't invent fMRI or DTI. They were invented in the 90s, and there's over 550,000 papers, and probably many people, if not everyone on this call, has heard about these things. So they're not new. So why aren't we doing this on everybody who walks in the hospital? It's not controversial that these things provide value, and it's fairly simple. There are limited software solutions that are able to do this end to end if you're not a coder or have a coding background. It's logistically painful to get these kind of scans done on a regular basis. So if you're at a place like Stanford or UCSF, you probably have someone who will do this for you, but again, they can't do it on a 30-minute of notice. They may not be in town, et cetera, et cetera. These are things that are not scalable across medicine and certainly not scalable to normal hospitals. And so if it only works in the ivory tower, it's not going to change medicine very much. And so we looked at this and saying that we need to get everybody into a common framework so that what we're doing in the research world can scale and to help patients in a much quicker way than what people are initiating. But as a result, because we don't get this on a daily basis, doctors don't have experience with these techniques really, and they don't have actionable insights that they glean from them. So what we get are really pretty pictures that can serve as a poster. If you go Google DTI whole brain, you get some amazing stuff that you can put on your desktop. But again, if anyone can look at this and tell me how cognition's happening from visual inspection, then you're a smarter person than I am. I spent a lot of time looking at these diagrams. I actually do know what some of these fibers are probably doing, but it's not really telling us everything we need to know. And that's because if our brains were simple enough to stare at a picture and figure out what's happening, we would be too simple to understand our brains. And so we end up not really leveraging the techniques and studying the brain the way the brain really is. And I want to show you the cost of that. Okay, so this is a picture of my wife on the left and Grammy Award winner Brian McKnight on the right. And if these two people both showed up with the same disease state in a current way we run trials, they could be randomized against each other. But of course, we know that there's differences, significant differences between these two people. You know, the genome is obvious, but also it's almost certain that their connectome differs in ways that are probably meaningful. And again, if we're testing things that have neurological impact in any way, then or does it really make sense to randomize this? And are we setting ourselves up to fail unless a drug or treatment is so beneficial that it just overwhelms the variance because it's just the cure for the problem? And there may be drugs that are useful that for the person on the right that aren't useful for the person on the left. So we end up doing trials that look like this. But reality is our patients come in like this, and this is actually what we're doing. We're studying things with vast amount of heterogeneity and hoping that randomization can clean this up. And given the challenge of running a 200 person per arm study, which many people know, this is probably not capable of cleaning up with randomization to really emphasize a drug that may have a 5% benefit and be useful. So again, what I would argue is actually a better way to do this is to really look at these patients, to understand them and create controls for them using machine learning such that we actually can study that and also many other subsets of that. And that's something that's more powerful. Now, again, that is one possible application of the Infinitome concept. It's a concept ultimately. This is what underlies under the hood of the engine. We take a standard atlas. This is what many people do if you're sophisticated, registering an atlas using volumetric methodology. One of the things that I found was not very scalable is that if there's been a stroke or a tumor or anything changing the shape of the brain, even Alzheimer's, this atlas doesn't fit very well. And it also might, because it's generic, it's not really, we're trying to take a round peg and put it into a square hole. And so what we do is we take this DTI or CSC tractography, and a machine learning tool looks at every voxel and reassigns it to an area that fits the structural connections better. So it knows, for example, the machine learning knows where the motor cortex is or Broca's areas based on who it talks to. And then we utilize this atlas, which is personalized to the patient, to subset the resting state and CSD tractography data to make connectivity matrices of all types. And what you can see here is this is a comparison of a bunch of areas of a large-scale brain network to a bunch of the same areas. So every little square is a comparison, and color is showing how much they're connected. In this case, how often they're firing at the same time. But it's, again, this is very nice little pictures. This is actually useful when we start to compare this against individuals, utilizing machine learning to tell us what is abnormal on the matrix. And again, what abnormal is a very complex term. But let me kind of take you through what that's doing in a little more detail. So let's say I have a brain and I've made an atlas. This is the Human Connectome Project Atlas. It was published in Nature in 2016, and they show that the cortex is not Brodmann's 47 areas. It's actually probably closer to about 180. And that, again, you can debate the exact number, but that's a pretty good map to our opinion. If you read the methods, they're very robust. And what they did was they compared you can take, once you've atlased the brain, you can compare every area to every other area. You look at the bold signal in each area, and you compare, do a correlation. You see how often are they firing at the same time. So a perfect correlation, which in that graph I showed you would be in red, is that these two things are identical. And a perfect anti-correlation, meaning -1, is that if this one's up, this one's always all the way down. So when you do that, you generate, in this case, what is a 143,000 square comparison, where every square on here is comparing one of these areas. You can see how many areas we're actually mapping. And the red squares are areas that are talking to each other, and basically, and the blue squares are the areas that are actually opposite each other. Now, in order to really leverage data of this quality and comparison, you need some complex machine learning. Now, as you can see here, what we do in order to make this simple, because again, if we have to manually upload thousands of scans or 30 scans a day, this is never going to happen. And anybody who's worked with this kind of project knows that things like centralized data repositories, data handling, de-identification is a huge labor-intensive solution. And that doesn't make any sense. Engineers can actually engineer that away from us. So we basically engineered that out of the equation, where we're actually able to put a gap server into a hospital that the scan can come straight from PACS with no de-identification whatsoever, straight from the MRI scanner even. Eventually going through a de-identification step and going into the cloud where the processing happens, storage happens in a bucket that is basically HIPAA, GDP, and compliant at high levels of security standards, but allows the doctor to call back the image and reunify it with the PHI on the hospital side. Now, if you think about how the method of how to re-identify that without us actually having the name here and without having it be traceable is a severely sophisticated set of engineering. But again, this is what we do. And so again, being able to get data in and out of the system is critical. Now, once you have data in the cloud, in a hospital, for example, would have a bucket in the cloud, and we don't own the bucket. We actually view ourselves as kind of a storage facility in that regard from a legal standpoint. What we actually can do is then launch large-scale machine learning algorithms on this. So it is actually possible, though again, nobody's attempted this yet, but it is possible to go analyze the data that came off these 1 million scans and tell me what you think is causing people's right arm weakness. It's possible to do that now. But the issue is you have to know who has right arm weakness. And so we started saying, okay, it's not enough to just be good at processing scans and getting things into and out of hospital systems. We need to have a way to mine medical records at scale. So what we did is we created a natural language processing platform that's a artificial intelligence modality that's able to read documents and figure out what they're saying and extract data from them. And if you've ever had any experience with this is a rule engine. And the rule engine here is actually based on how I would read a medical record because I helped the developers build it. And I've written a lot of clinical research studies if you go look me up. And what we did was we said, first, take the name off of it. We can identify names and take them off with very, very high accuracy in multiple languages, including Chinese. And what we then can do is go in and look at the notes and say, yeah, we don't really want those notes. They're not that useful for research and they create noise. And then if you're reading this kind of document, which is an H&P, go find the subheadings, keep the data, make sure you keep track of where you got the data, read the medical record and extract medical terms, make sure that you handle negations, and that, for example, when you assign a diagnosis to the patient, you look at the assessment and plan because that's probably a lot more accurate than other parts of the medical record for getting that. Also, it's a significant interest to know where cognitive functions are in the brain. So if someone gets worse on a neuropsych testing, instead of locating to the parietal lobe, we'd like to locate it to a tract or a parcellation or a network or all of the above. And again, a couple people pointed this out to me, that this was even at places that had phenomenal neuropsychologists, it's not a scalable solution. They're busy, they teach courses, they've got research, et cetera, et cetera, et cetera. And so if you can't get a 5-hour neuropsych test, which is on probably 99% of individuals in your hospital, can we find one that's quick, scalable, and can be administered by non-experts and utilizing the computers to do the scoring? So of course, the biggest barrier to that would be aphasia. So of course, it's not intuitive that a computer can listen to language and tell you when it's abnormal. But if you were to take something, have it come in as voice to text, and then turn loose something like a deep learning algorithm on it that's trained to recognize and classify types of aphasia, it's theoretically possible. And our prototypes show that we can do this about 92% of the time accurately now. And so if we can score aphasia, everything else is fairly easy because, again, voice to text is fairly good at matching words. And when you start talking about anything else, it's fairly easy to build rule engines that can score things like this. And so when we put all of this into one container where you link the medical data to identify the patient, identify confounders like other medical problems, link it to Connectomic data, it allows you to go in and have powerful algorithms that you can launch to identify that in this one patient history, that there are actually two scenarios with two scans that you can analyze different scenarios to try to figure out, okay, instead of having to go find the 400 recurrent glioblastomas that have treated at your hospital in the last 10 years, you just use the software to do this. And building a query tool that non-experts can use is challenging, to say the least. But you can see what our prototypes look for, and we think we have a good solution to this that should be in Infinitome in the near future. So once you've identified these people, then it's fairly simple to just say, launch a very sophisticated query. And artificial intelligence and data science and machine learning is really more about asking good questions in terms of machine learning problems than it is about actually coding these things nowadays. Nowadays, these are a couple lines of code if you have the way to implement them. And once you can implement them where these are modules, so you just say, run this operation on this data set, now what you have is a way to mine your own data for big data insights. But where it really becomes extremely powerful, because again, theoretically, if you had some people on staff who are really smart, you could probably do some of this yourself. It would be challenging, you know, but where it really becomes a benefit even to sophisticated groups with those kind of people on staff is, let's say we have 3 hospitals here in 2 different countries and they want to do a research project together. Well, there are rules about transferring medical data across international lines and where the cloud data is stored. There's also challenges with regards to auditing, you know, so if you're doing a large study and this is going to ever go in front of the FDA or ever go in front of critical peer review, it's generally thought that we need to have groups auditing in. This involves this large, long process of going through and finding physical charts and linking them to data in the database. And this is a huge amount of time and effort. Forget the fact that someone had to enter all this. But if you do all this in an automated cloud way, what you can do is you can just send the algorithm out to all the buckets. Just tell us, give it an idea what you want to identify. It does what's called federated machine learning, where it runs the algorithm in the cloud where the data is located and brings back the weights, which actually isn't, you know, doing anything that anyone has a problem with. And you can then combine them back at the mothership to have your model. So this allows a bunch of groups to work in a way that's auditable, traceable, and simple at a substantially reduced cost. So we looked into this in some of the projects that we're helping to organize, and this can oftentimes reduce the cost of doing a very big, hairy, audacious project from several $1 million to a few $100,000. And of course, anyone who's applied for grants knows that which one is much more likely to get funded. And so if you were to look at this, this is the kind of project you could do where you have countries that don't see eye to eye, to say the least, and you could theoretically get the doctors to cross those international lines and work together in a way that is safe, traceable, and effective to really handle large-scale connectomic projects with high-level, high-quality data. And again, then you'd think, what does a machine learning model look like in the connectome? And the answer is, it looks like a tool, a visualization, or some kind of classification of data. So if you go back to the viewer, what we're trying to create ultimately is, where do you look in the connectome? If someone has a language problem, where do you look? Well, you can look at the language system. We have that programmed in there. But there may be other areas that cause other types of language problems. So where do we look? If we're diagnosing Mr. Johnson, who has a language problem, where do we look? And that's what we're really trying to do, is to provide visualizations that have clinically actionable endpoints. And so what I hope I convinced everyone is that it's now possible to turn this that hospital into a data machine and really began to do research at massive scale to bring the kind of complexity to the data that we need to really handle these problems. So thanks, everyone.
Can you all hear me out there?
We can, yes.
All right. Thanks, Mike, for that introduction. And I am by no means what I would consider expert in human connectomics, but very interested. Probably the one of the most exciting things I've seen in the last 3 years for sure, and one of two in the last 5 years, as things would, with respect to applications for neurosurgery. This is my disclosure slide. I think it's kind of interesting about how things evolve, and this is the way Charlie Wilson said it should be. And that is, he said to me when I started in 1992, he said in his Louisiana accent, "Michael, it's my job to make you famous. And if most of the faculty at UCSF end up being program directors or chairman, then I'll have done my job well." So the history between Mike and I is that, you know, I was one of his teachers and he was one of my students, and now it's reversed. Mike's a professor, he's CEO, co-founder of this company, and he knows all about Connectomics, as he's phrased it. He's the teacher and I'm the student. I first became aware of this at one of our national meetings, I think the CNS meeting, where the Congress of Neurological Surgeons published a supplement free to all residents and produced in hardbound copy as well as PDF format, along with a Toronto-based company, about what Mike called the Connectomic Atlas of the human cerebrum. And chapter 1, and I believe there's 18 chapters in total, was an introduction to the methods and importance of this Connectomics project. And with all respect, even though I found it very exciting to read, it was mentally exhausting to try and get your head around some of the concepts that were presented in that article. But these were the regions that were divided up for some of the initial analysis involving human brain specimens. And rather than me discussing it, it's best that you just read the article. All articles are available through PubMed, open access, downloadable as PDF files for everybody to use. And originally I remember having the book about by Brodmann, discussing the divisions based on classic neurology, which means history and physical, because there was no MR back in his day. About 47 areas, and some of which were most importantly related to language and motor function. And some of these areas were, of course, confirmed and supplemented by the work by Wilder Penfield at Montreal Neurologic Institute. But I wasn't aware that there was a large-scale, publicly funded, multi-institutional effort supported in part by the NIH called the Human Connectome Project, which converted the standard classic 47 divisions, also called parcellations, into a much more complex classification of 180 different segments, which is what is referred to in Michael's introductory chapter on the methods. Now, as others have pointed out, the brain is an electrical organ. It's not a static piece of tissue. It's not tofu, even though it feels like that under certain operative conditions. And, you know, Al Roden, I used to call it the crown jewel of human creation. And even I think Mike would say that at this point, even artificial intelligence doesn't have the flexibility. It can compute a lot faster than we can, but it's a little more rigid than the human brain. But what Mike pointed out in that first chapter was that brain networks follow a, quote, small world pattern of organization. So you can look at the U fibers, association fibers, longer communication fibers in the same hemisphere, communication between hemispheres, same regions, different regions. And this was apparently first published back in the 90s by two authors, Watts and Strogatz. And I think the effort that Mike was trying to make was to use big data to reduce the complexity of the networks of the human brain and to try and display it in ways that we can better understand. It turns out that complexity is governed in most natural systems by common mathematical principles, but you need large amounts of data, complex parallel processing, and machine learning to really get the most bang for your buck. Most neurosurgeons know what these are, DTI images. Most of us would be able to point out the uncinate fasciculus, descending motor fiber pathways, Myers loop, etc. But to be able to understand everything that's in the images of A, B, and C is an extremely complicated problem, even for neurosurgeons that routinely operate on intraaxial tumors. And if I can dumb it down a little bit using some of Mike's video information, as you've seen on his Infinitome machine, he's basically combining 2- and 3-dimensional information from standard MRs, fiber tracking, and a resting state functional MR, analyzing that data with machine learning to produce connectomic brain maps so that we can provide better care for our patients and possibly assist with using this technology for more sophisticated and specific diagnoses in the future. Anybody who's seen one of Mike's seminars online, I mean, I was blown away by it. Every person that I've referred to sign up for one of these seminars, equally impressed. And it does take a couple of rounds to really completely understand the concepts and the functionality of the system, but I think it's truly amazing. I've never seen anything like it. And I was very excited, and hence my involvement and interest to participate. So we're a simple shop. We're affiliated with Florida International University, but this place was largely private practice until I arrived. What we did with Michael and his group was to pay for the analysis of 250 scans into 6 different projects. We didn't know exactly what we were looking for, and we're going to look to Michael and his colleagues to help us with that. But we'd like to be able to add our data set to other data sets so that we can increase the numbers and then analyze and get more out of it. Even though we don't know what we're looking for, I think there's certainly an excitement about the technology. We're discovering, we're collaborating, as Mike showed, internationally. I think it's awesome. The issues related to uploading of MR imaging data sets and personal health information firewalls, et cetera, has been solved. As Mike showed there, I think it's an ingenious idea. And even at Baptist Hospital, which is extremely conservative, I mean, they've got firewalls for everything. You can't even download CVs of applicants from a commercial processing site because they're so restrictive. But our IT department has ultimately agreed that everything we're going to do with Michael and his group is sufficiently secure that we can proceed. So what I arbitrarily decided was I had 250 scans and I separated them out like this. My colleague, Edward Chang, who's now the chairman of neurosurgery at UCSF, does 1 to 2 epilepsy operations a week. And so he's got a wealth of experience and can analyze these patients prospectively, both before and then after. So if he has 60 scans, he gets 30 patients Baptist Stroke Program, 750 strokes per year system-wide. Philippe de los Rios is a neurologist who works with us. He's the director of the stroke program, so he gets to analyze 35 patients. I took an interest in NPH because none of my adult colleagues knew anything about shunts. And so I ended up defaulting into an interest in that. And so I've got 15 patients I can analyze pre and post shunting when they respond to provocative testing. We'll look at adult brain tumors. We'll look at our own small epilepsy program here in order to stimulate activity and interest. And then we have a neuroscience group working in the fields of memory and aging. And in particular, they have expertise in transcranial magnetic stimulation, which Michael has shown us may have utility for treatment and promoting recovery for patients post-stroke, post-surgical intervention and injury. So when I started to look at the Connectome coordination facility, which Michael was referred to as a multi-institutional site, it houses and distributes public research data for a series of studies that focus on connections within the human brain. And I looked through a number of the projects, and here just on the right-hand side, you can see some of them related to resistant depression, Alzheimer's disease, aging brain and dementia, epilepsy Connectome project. A number of these sites happen to be in North America, but as Mike showed, we can be anywhere in the world and combine our information to promote neuroscience. So as a bread and butter brain tumor surgeon, I think this is one of the most exciting things I've seen since September 2016. It's a new method for analyzing brain structure and function pre and post intervention, and expands the potential role and indications for transcranial magnetic stimulation. I also think, as Mike has shown in some of his seminars, there's a big area for advanced and more specific diagnostics, as well as refining our intraoperative techniques. And I thought it was really interesting in one of the seminars, Mike showed a subtotal right frontal lobectomy because the medial aspect of the frontal lobe up against the falx had considerable posterior connections, which were preserved by simply removing the abnormal tissue on the T2 weighted image rather than just completing the lobectomy right to the falx and have a more impressive scan. So hopefully this kind of information can help improve patient outcomes as well with surgery. So that's all I have to say, and thanks for your attention.
All right, well, you know, these are two very hard acts to follow. You know, you've heard from two wonderful world-renowned neurosurgeons. I'm a behavioral neurologist, and so I'm going to provide a more focused application of the Infinitome to neurodegenerative disease, which is what I commonly see in my clinical practice. So I'm based in St. Paul, and where I work is the Health Partners Center for Memory and Aging. If any of you happen to be in town, please stop by. In the upper left-hand corner is our new building that just went up in 2017. It's overarching Neuroscience Center. So, you know, I just want to set the tone about the disease that I see most commonly in my practice, Alzheimer's disease. Every year there are these facts and figures that come out from the Alzheimer's Association. We know that it's the 6th leading cause of death in the United States. It affects 5.8 million individuals in the country, and it's fairly costly. It's about $305 billion a year for the care of these patients, and it's expected to increase to 1.1 trillion by 2050. The pathological signature of this disease is characterized by these amyloid plaques and tangles. These plaques consist of beta 42 that form outside of cells, and then intracellular deposits of these tau inclusions that come together to form tangles. Now, these two pathological entities compromise various large-scale brain networks, and you see that in this diagram below. And as you have progressive network failure, you end up with neuronal death, and as a result, you end up with the end stage that you see in the right-hand corner, progressive brain atrophy, and particularly involving the limbic structures such as the hippocampi and medial temporal lobes. Now, one way to make some of this research relevant is to think about the patients that we see every day. And as Mike Sughrue said, go where the disease is. And so this is a common patient that I see in my practice, a 73-year-old man who comes in with 2 years of memory problems. He's repeating himself, forgetting conversations. He was an investment banker, and now he's struggling to manage the finances in his home. And he does cognitive screening that is abnormal, and then undergoes formal cognitive testing, which in my institution is actually not too hard to get, and has the characteristic deficits in learning and memory. And then we perform the brain imaging, and often the families are hoping that the brain imaging is going to show us exactly what is going on in the brain and allow us for very specific diagnosis. And what do we hear from the radiologist? Well, those of you who treat these patients are probably very familiar with this reading of age-related cerebral atrophy and microvascular disease. And often we're using these images to rule out structural abnormalities like stroke or tumor. And so by exclusion, we make the diagnosis of Alzheimer's disease. And then the common question we get, well, does my husband need repeat imaging in about a year to track the disease over time? And often I'm telling them that we don't have that sensitivity to do that with structural MRI. So I don't know if any of you are familiar with the movie The Matrix. It's about this man, Neo, played by Keanu Reeves, who lives in this reality, which is essentially an unacceptable reality. And he has to choose between taking the red pill to come to terms with this unreality and to change it versus the blue pill to remain ignorant and blissful. And to some extent, we neurologists are facing this same dilemma. We live in an unacceptable reality where we have been taught that the MRI is where the buck should stop, and that's not the case. And I think since all of you have signed on to this webinar, I'm assuming that you all would choose the red pill. And I'm going to give you several examples here where structural MRI just fails us miserably. I asked you, what is the characteristic pattern of abnormalities in Lewy body dementia? Or what about that patient who comes in and has been admitted for sepsis, and on day 14 of the admission, there's a neurology consult where we're asked to evaluate persistent encephalopathy. And we do an MRI, and it really doesn't show us anything that's abnormal. Or what about those patients that Dr. McDermott referred to with normal pressure hydrocephalus? And we're trying to struggle between determining if they have a progressive neurodegenerative disease, in which case you don't want to shunt them, versus a patient who would benefit from VP shunting. And so, our MRIs give us very limited information in these cases. And as Mike Sughrue alluded to, our brains are much more complicated than what we see on the structural MRI. It's more than just a bunch of gray matter, but you have these billions of tracks connecting various structures. And essentially, we end up with a variety of connectomes, variety of networks that are communicating with one another to serve certain purposes, whether it be language, memory, executive function. And this level of analysis and understanding is what's necessary to be able to treat cognitive and behavioral diseases. Now, the Infinitome program is really exciting because it's able to take these billions of connections and simplify it and allow us to look at various networks, such as the default mode network, which is commonly affected in Alzheimer's disease, and analyze them. And that's one of the ideas that really grabbed me when Mike first told me about this program he created. In my field of work as a behavioral neurologist, we're very interested in monitoring changes over time. The idea of biomarkers is something that we talk about and focus on at our annual dementia conferences. And there are a variety of biomarkers that have been studied. And I think that resting state functional MRI, by leveraging the Infinitome program, will probably be a new biomarker by which we gauge progression of Alzheimer's disease from the mild cognitive impairment stages to the mild, moderate, severe stages of the dementia itself. So I already talked about the limited use of the structural brain MRI. We also frequently perform brain FDG PET studies where you characteristically you see hypometabolism involving parietal temporal regions or the posterior cingulate cortex. At the same time, these changes may be present as early as the mild cognitive impairment stage and may not change significantly until you get to the latter stages of the disease. In addition, the specificity of FDG PET is somewhat limited. It ranges from 50 to 71%. And so there is more work that needs to be done, and it's not a perfect tool. Now, over the recent 15 years, there's been the development of molecular markers. Essentially, you give a ligand that binds specifically to the pathological markers, whether it be amyloid plaques or neurofibrillary tangles in the form of tau in a living individual. And this is very exciting for diagnosis, but likewise, these biomarkers are not perfect. First of all, when you look at amyloid PET and you give this ligand, you have a positive study, there's no clear correlation between the presence of amyloid plaques and the disease severity. So this may not be the best way to track changes over time. Also, at my institution, it's extremely costly study, about $13,000 per scan. Now, tau PET is a more recent imaging modality, but recent studies have shown that the effect size for longitudinal change is actually not superior to cognitive testing, which is readily obtainable at my institution. And then if you look at some of the CSF biomarkers, and certainly we can do spinal taps on patients, it's more invasive than imaging, but the studies looking at autosomal dominant Alzheimer's disease and also sporadic Alzheimer's disease have shown that these biomarkers do not change significantly over time. And there's a more recent study looking at longitudinal change in tau and phospho-tau, which should correlate more with clinical symptoms. Well, these change minimally over time in Alzheimer's disease. So if we think more broadly and we put together an idea that the misfolding of these proteins, the amyloid protein and also tau, and how it leads to clusters of amyloid plaques and neurofibrillary tangles, and how that interfaces with large-scale brain networks, I think this is a better way to understand the progression of Alzheimer's disease. So you end up with these pathological entities, you compromise large-scale networks such as the default mode network, and then as a result, you end up with network failure, and then further production of amyloid plaques, and you end up with a vicious cycle. So the way the pathological entities interact with the large-scale brain networks is probably a good way to think about this disease. And utilizing resting state fMRI may be a more sensitive tool to track changes, longitudinal changes in neurodegenerative diseases, such as Alzheimer's disease, frontotemporal dementia, Lewy body dementia, what have you. And some of our work is focusing on this very concept. So one of our recent projects is looking to correlate large-scale brain network dysfunction with disease severity throughout the Alzheimer's continuum. And our second aim is actually to look at neuropsychometric abnormalities and then correlate those with dysfunctional networks and also nodes within those networks. So we are interested in the default mode network, which is very well studied in Alzheimer's, but there are some other frontally based networks, such as the salience network, which is responsible for maintaining attention to emotionally salient stimuli, and then the central executive network, which is responsible for maintaining executive function and performing cognitively demanding mental activities. So we believe that these networks are all affected in Alzheimer's disease and may be progressively impaired as the disease progresses. So one of our outcome measures is using and leveraging the Infinitome software to look at connectivity within large-scale brain networks. Our institution does not have 4 fellows to analyze this data, and so it really makes sense for us to collaborate with Mike and Omniscient and use Infinitome to better understand these areas that are dysfunctional. So we have these connectivity matrices that can tell us specifically what areas, what structures or nodes within a specific network are dysfunctional. And by evaluating this in comparison to human connectome data, we can see for sure if there's abnormalities or if there is intact connectivity. So using this legend here, if you're in the blue region, that means that the connectivity is compromised, and you can also look at the degree of standard deviations from what would be considered normal. And so this is how we may hone in on some of these connectivity anomalies we may find in large-scale brain networks. The second aim is to look at various cognitive presentations and to specifically look at cognitive modalities that may be compromised and link those to degenerative large-scale brain networks. So for instance, we hypothesize that learning and memory will co-localize or correlate with the default mode network dysfunction. And then salience network, which is responsible for attention to emotionally valent stimuli, that should correlate with attention. And then lastly, executive function should be associated with the central executive network. And so patients who have executive dysfunction, we'd expect that to also correlate to further anomalies within the central executive network. And so these are kind of the ways that we hope to decode and better understand the progression of Alzheimer's disease and the compromise of large-scale brain networks. And this is something that's applicable to all types of neurodegenerative diseases. And I think it's a very exciting time to be in this field. And I thank you for your attention.
Nice presentation, Mike.
Yes, great. That was 3 excellent presentations. I want to thank our 3 speakers, Drs. McDermott, Rosenbloom, and Sughrue. As we enter our Q&A session, I would like to remind everybody that they can submit questions using the Q&A window in the Zoom link or in the Zoom interface. It says Q&A, and you can type the question there and we'll receive it and we can ask. And we'll go ahead and launch 2 more poll questions while we're waiting for question submission. So go ahead and answer those. And while we're waiting for questions to go in, I might go ahead and pose one. I think this is a great approach to collect data and understand, and collect the data and then ask some very interesting questions of the data. But I'm kind of curious from the clinicians that have put this in practice, what is needed from an IRB perspective to get this type of study up and running? It's a bit different than a normal study.
Maybe Michael has more experience, but it took us 3 months through our research office and IT, as Corey and Brad and Michael know, to get approval for the uploading of MR image sets for analysis and cooperation with the federated site, but it's doable. So I don't think it would be, it's not that difficult, but if you have support staff, administrative staff who are interested and can help work with your IT department, it can get done. No problem.
At our institution, it involved me interfacing with our IRB, and actually they, our application sailed through it. That was actually the easiest part for me. I found it most complex when I was working with our IS&T department. And
Yeah.
there are various, legal representatives within that group, and safety and security. And we had to pass through quite a few hoops. So that took, I think, likewise, at least 3 months. And then also I work with our neuroradiology group, and they had done only limited resting state functional MRIs, usually for neurosurgical patients. And we were talking about doing research that would involve, hundreds of patients. And so we had to come up with some type of standard order and protocol. And fortunately, we had Corey Dixon working with our various teams. But I would say IS&T was the toughest hurdle to get over for our institution.
Great. And I have another question. One thing that strikes me is, as you were speaking about the possibility of different biomarkers, this seems like it would be of interest to pharmaceutical companies. I'm kind of curious on your view of how this type of technology or platform or imaging technique might get involved in those types of studies going forward.
Who's that question directed to?
Let's go to Dr. Rosenbloom.
Yeah, so I think there's a lot of opportunity for clinical trials. We at our institution, we've done some physician sponsored trials where we made our own design. And I think when it comes to looking at biomarkers over time, I think one of the points of my presentation is that this may be a better way to track progressive neurodegeneration over time. And for reasons that Mike Sughrue explained in his presentation, the functional MRI is a very abstract concept to many people who are in clinical practice. And it's really difficult to do the work to utilize it for clinically meaningful purposes. And if we can outsource that work to, Omniscient through Infinitome, that makes our job much easier. So I think this technology, by leveraging this technology, this would allow resting state functional MRI to play more of a role in clinical trials. And right now, for dementia anyway, we're looking at the very biomarkers I described, MRI scans, FDG PET scans, CSF biomarkers, amyloid. But it would be nice to be able to look at something that has a one-to-one relationship to the clinical phenotype. And it makes sense to me that the network that is being degenerated in the disease process will most likely relate to the symptomatology. I mean, as neurologists, we like to link clinical presentations with the regions in the brain. And when it comes to cognitive behavioral conditions, these are networks mainly. And so the question is, what network serves the memory that is compromised in Alzheimer's disease? Or when patients progress and they have language and executive dysfunction, what other networks are involved? So I can see this as being applied to clinical trials, and Infinitome, I think, simplifies the process of utilizing resting state functional MRI.
That's great, thank you. So we have a few questions from the audience. I think these are both geared towards Dr. Sughrue. So the first question is, can you address the ability of the software to integrate new OFD data that we're replacing DTI? I'm not exactly sure what OFD is. Maybe you are.
I think he might be talking about NADI and similar sort of cue ball. So the answer is we can integrate multi-shell. There's always, there's millions and millions of potential algorithms that people could put in there. It will generally handle them reasonably well, but some of them we're probably not going to do all the justice that are possible to some of these algorithms. I mean, again, there's diffusion, relaximetry, and things like this. Ultimately, if someone's doing something really, really complex, it's not really in our wheelhouse because most other people aren't going to be doing it, and we need to have something that can scale. And so that's what I would say is that anything reasonably achievable in a non-research world setting, we can probably handle. but there's always there's people making their own, PSG files for scanners. Again, that's all great. And if it starts becoming scalable, we're happy to work it into it. As far as 7T, as long as it's compliant with the DICOM standard, we have a unique way to fix gradient distortion. That's the real challenge with 7T. That should scale to that, but we haven't specifically validated it because there just aren't that many 7T MRIs out there.
I would be curious to to speak more about that. You can contact us at that contact@08t.com email. We can take a look at the 7T data. So moving on to the go ahead.
Sorry, go ahead. I mean, the answer to it is we've got a lot of very smart people who can figure things like this out. One of the issues then, of course, is always as a small startup company, how many edge cases can you really tackle? As far as the next question, how do we know that the correlations are true biologically? Now, what I'll say with this is that ground truth in anything in the brain is very difficult because we cannot get an entire mapping to EM space. It's just not possible to map the whole brain with EM. To get put in perspective, the manpower in order to map 100 nanometers cube of EM is like 10 to the 9th man hours. And I think they've got it down to 10 to the 4th recently or something. But that's not something that's scalable to the whole brain. And so we don't have a ground truth map. And even if we did, we'd have it on one brain and not necessarily on the brain that we're studying. And this is a epically challenging problem. What we'll say with it is having done, and you can look at, we've published a small library of tractography compared to manual tract dissection. And grossly, we know that anything we find in the manual tract dissection, we can usually find in the tractography. Neurotractography isn't using isn't looking at a very minuscule set of data. But inevitably, there's a degree of randomness. And so if you run the algorithm several times, it's possible that you would get some different results. But we've actually looked into this. And what we've done is we've repeated it over and over again on the same exact data set, and we get 99% overlap between tracks, and we never really see voxels get reclustered in our algorithm by doing it twice. So it's stable. And so what I can say is that, like any tool in any methodology, you can't trust it off the cliff. It has its limits, but it's pretty good. And again, if you go look at individuals, you see a lot of the same patterns come up over and over again. One of the challenges is, and again, it's an epically hard challenge, of how do we do this in a way that actually adjusts the fact that everyone's brain is a little different. And this is what machine learning is all about. Now, the issue with it is getting specifically to our atlas. What we looked at is we said, are we utilizing the machine learning to replicate Glasser's atlas? And we thought about it and talked about it. And finally, someone just pointed out, well, no, actually, it can't possibly be Glasser's atlas. It's going to be very similar to the HCP atlas, the Human Connectome Project atlas. But it's going to be different because we're basing it on tracks and not on position in space. So it uses similar nomenclature. And in a normal person, we've studied this, and it moves about 2% of the voxels. So, and doesn't do anything really weird like it moves it to the other side. And the answer to it is that it is a similar atlas, but it is different because it's a tracked atlas. It's a track-based atlas. Now, one of the questions is then what we should say is, how do we know that it's accurate? And the answer is, you think, what do we really look at? And so if you had a surrogate that was telling you something biologically inaccurate, but it always predicted a clinical outcome that we care about, do we really care if it's biologically accurate, if it's telling us something that is repeatable? And we've kind of angled around a different strategy to say, what we're going to say is, we have an atlas and it behaves properly and it's stable and it's biologically plausible. Let's figure out what it's telling us in terms of clinical medicine. As far as does the right IFG correlate with the left IFG, it turns out you happen to pick an interesting part of the brain. So a lot of times symmetrical parts of the brain correlate because a lot of networks are bilateral. There are actually 5 parcellations in the entire brain that are parts of different networks on different sides of the brain. And one of them is area 44, which is Broca's area. It's in the default mode network on the left, roughly, and on the central executive network on the right, because language is a DMN function. So Turns out if those are correlated, then it's not an error. It probably is something pathologic. What I'll say from as far as the accuracy is that when you take something through the FDA, which we're in the process of having some of our algorithms go through the FDA right now, you end up having to do a gigantic mountain of validation and unit testing. So what we've done, really is we have tested all the aspects of the code. We've run it against numerous algorithms. We tested against other people's algorithms. And I personally, who probably in my life have looked over 2,000 DTI scans of various kinds, have looked at our algorithm hundreds of times in pathologic states, and we know that it's not doing badly. But then, of course, what you get down to is radiological images to go down to absolute certainty is ground truth. And in the brain, when you're talking about this level of resolution, we don't have a pure ground truth. And so one of the things we thought is, we know that our algorithms perform as well as anyone else's algorithms. They're very good. And they seem to be behaving the way we would expect. What we then say is, given that these are reliable, whether they're accurate biologically is a whole different question. Let's try to figure out what they tell us clinically. And if it turns out that there's a slightly better model that's possible, we'll adopt it if someone learns it, but that doesn't exist right now. As far as the neuropsychological testing, I'll answer that question too. We have a variety of tests that cover a cognitive domain. This was worked out with talking to several neuropsychologists, including people at the Mayo Clinic, MD Anderson, and several other groups, including having one of our lead developers have a significant background in neuropsychology. And we came up with a test that can be done in about 45 minutes. It has things like familiar versions of things like Trails, Hopkins Verbal Learning, the MMSE, the Corsi block tapping, an aphasia battery, not Western because it's just too long, and a few other ones I forget off the top of my head, the Benton visual memory test or something. And so what we did is we took those tests and adapted it so it could be run through an iPad or potentially through a web browser. In the era of COVID, that's really important. And we aimed to make it so that it was somewhat self-explanatory, such that you didn't need to have a neuropsychologist doing the test. And so we're in the process of collecting a large amount of normative data on this and making sure that we have that normed, so it will be normed in the software. But the whole goal is that we need to get the kind of process where it doesn't require a PhD to do this. Otherwise, it will not scale. I hope that answered the question.
Great, thank you. And we're now 15 minutes over time, so I'm going to go ahead and wrap this up. We are happy to answer other questions. If you have them, please submit them to our contact email address, and we'll get an answer back. So thank you again, and thanks again to our three excellent speakers.